Okay, so good morning, everyone. So today I'm just going to finish going through the whole coalescent theory a bit. I'm going to talk a bit about what happens when how you can think about selection in the context of the coalescent model. And then I'm going to go to talk a bit about data and what people actually do with this stuff afterwards. So, so selection is very difficult to model backward in time the way you do with a standard coalescent, because the whole trick here, right, is that we can separate state from descent, right? You can figure out the lineages of descent, and then you figure out what kind of allelic state they have, whether it's dropping mutation on a tree, right? And that only works because reproduction is independent of what particular allele you carry, right? And selection, by definition, violates this, right? That's what selection does. So there are two different things that people do, and one is what I usually think of as a conditional structured coalescent. That means that you think about a particular selected locus, and you sort of condition on what happens at that, and then you model the genealogy conditional on what happened at the selected locus. So you kind of go both forwards and backwards, and I'll explain that in a second. Then it is also possible to formulate a coalescent-like model, like an ancestral selection graph. And I'll try to just go through that very quickly. And this is not something I thought about for a long time now, so bear with me, see if we'll have to remember it together. This actually gets pretty complicated. And there was great interest in it 20 years ago, but people haven't done very much. It's very, very theoretical. So the ancestral selection graph, I'm going to try to do this on a very intuitive level. If you really want to understand this, if you like stochastic processes, right, there are some really cool papers on this stuff, but they're not easy, right? This actually gets sort of complicated. So what they do is that they decouple descent from state by introducing virtual branches back in time, sort of branches that could have existed, that could exist depending on what mutations end up traveling along them, right? Because selection in some sense means that some things that could have lived actually died, right? So you have to introduce these ghosts for, you have to leave room for selection as you put in the graph. So you do this thing, you add a bunch of weird extra branches, and then you put on mutations, and once you know where the good alleles are, you sort of kill off the branches that happen to have the unfavorable allele in a sense. So that's the intuition. Yeah, so the easiest way to model this is using the Moran model, which is a model which I explained before. It's formulated in a very different way from the Wright-Fisher model, but actually in the limit looks exactly the same. It gives the same diffusion limit, etc., right? So in this model, you have n haploid individuals, and this is now in continuous time. And then you have some rate at which individuals give birth at a rate determined by their type, if you go forward in time. And this newly born individual then replaces somebody else in the population, right? So you have constant population size, somebody gives birth, and the offspring sort of kills off another lineage and continues that way. And then there's a mutation rate. Okay, so we're going to assume that there are two types, A1, which reproduce at rate lambda 1, and A2, which is slightly more fit, right? So reproduces faster than the other. Okay, so then the way Claudia Neuhauser and Steve Krohn formulated this, and like I said before, there has been more work on it. So my knowledge of this is sort of goes back to that time, but the literature gets pretty esoteric. They use the so-called biased voter model, which is basically a way to model how people vote, but with some kind of preference, right? So time goes forward like this. You trace lineages. Here is a population size of 8, and then you put down arrows at rate lambda 2, which is the highest of the two rates, and then you basically decide an arrow is green with rate lambda 1, so that's like the lower reproductive rate, right? So you go for here, so then you just throw in these arrows. Okay, so these arrows represent sort of putative birth events, right? Okay. And then you toss on mutations at the beginning here, right? Because now you put on the mutations. So remember, we do the same mutation dropping as you did in the standard coalescent, right? You just put mutations down the tree. So you put on mutations here, right? But a clear problem here is it's difficult to know where the stationary state is here, right? Unlike in the other one. So this is technicalities. It can be solved. Okay, and now, so the reds represent the A2. Those are the fitter ones, right? So they can use both types of arrows. They can give birth to both types of arrows. The white ones cannot, right? Because they're the less fit. So let's see what happens here, right? So if I did this right. So now descent depends on state. So if you follow the ancestry of this guy here, right? If this one is a red, here it runs into a green arrow. It gives birth, so it has a descendant that continues on that slot in the population, the end slots in the population, right? And it continues this way, and here's a red arrow, and it can give rise through that one as well, right? So basically, yeah, so I'll go back again here, see if I got this right. Okay. Right, but now, if you have we're going to shift this guy here to if this one here was a white guy, so this is the A1 type, again it gives birth here, but it cannot give birth through this branch here, so it doesn't get to reproduce over here, right? So in the end, you get a different, if you now go back and follow the genealogy, let's see, how did I do this? Okay. So if you follow the genealogy back here in time, now the actual ancestral tree will look different depending on what allele you had in the beginning here, right? So, and when you work this, if you work through the math on this process actually describes then the genealogy of these different branches, right? There's basically more opportunities to give birth through if you're the fitter type, right? So basically, what you have to do here is that you have to throw in all these virtual splits, right? So you can't just follow the lineages back in time, and like you did in the standard coalescence, and just see who coalesces, and then drop on the mutations, because who coalesces with whom depends on what state they were. So instead, you have to model it. You have to put on this graph-like structure, then you put on the mutations, and then you sort this out, right? And as you can probably see, I mean, if selection gets strong, right? What you end up doing, you end up throwing in a ton of extra branches that will never get used, because only like the fit alleles can get through there. So like all the deleterious alleles will cause, the more selection there is, the more of these ghosts there will be in the population. I mean, those represent things that were killed by selection going forward in time. Anyway, so that's how you can do it. And like I said, there is much more done on this, right? But it's almost really never used very much. So formally then, each lineage independently branches at the rate that depends on the maximum selective difference. Each pair of lineages coalesces at rate 1. So since you have these extra branches putting in here, but since the sort of selection is sort of linear, coalescence is quadratic, you're guaranteed that you're going to have some kind of finite number in this, right? In the end, you actually get a graph-like structure out of this. But just like with the ancestral recombination graph, it can get very, very big if selection is strong, because you have to throw in lots of extra branches. Okay, and so I think that's really all I'm going to say about that. I mean, if you want to read up on more of this stuff, right? If you just go and find that Krohn and Neuhauser paper and then just follow the citations forward in time, there is a literature on this, right? But there are very few people in biology who actually work on it, because it's too complicated to be very useful, right? And you can only model you can only model weak selection at a small number of sites, right? Which is not terribly interesting, really, in most settings, right? I mean, when we're interested in weak selection, then we usually think about many sites, right, across the whole genome, or we're interested in things like selective sweeps and stuff like that when there is a strong selection. And of course, you cannot do that with this kind of model. When you have something stronger, so then you can use this approach instead. So, right, You can sort of read for yourself. So basically the idea is we are going to think, if you knew the trajectory of the selected alleles, right? If you think about a gene with 2 different alleles under selection, right? If you knew going forward in time which trajectory they had taken, if you condition on that, then you can think of the population as being divided into 2 different classes, just like population subdivision. You either belong to the, this allelic class or that allelic class, right? A particular sample. And then you can do a coalescent backward in time in this way. So it looks like population subdivision, but mutation, you can only coalesce if, when there's a mutation between these 2 allelic classes. And in the end, if you look at a bigger region, you can also, other regions can recombine from one allelic class to the other, right? So how could you do this? Well, this may be reasonable if selection is strong, because then, sometimes the trajectory of the selected allele is actually deterministic. This model was invented for balancing selection, where people imagined that there were like 2 alleles that had been maintained at sort of constant frequency almost for a very long time. But you can do the same thing for like a selective sweep. You can like simulate, if you want, a selective locus sweeping through a population forward in time, fix that, and then think about the neutral variation here. And so why would you want to do this? So the idea is, again, that you have some kind of sequence data. And if there's like a small number of sites that are under selection, they will affect the variation at all these neutral sites around, which is what gives you the signal of the past, right? If there's just a single site, you can't really say anything about it. And I'll show examples of that in a second. Right, so you use it for detecting selection, right? Okay, so let's look at balancing selection. You have 2 alleles that are maintained at constant frequencies, P1 and P2, and mutation, they mutate back and forth. And now we're going to continue a segment, a chromosomal segment of some scale length, recombination length, around that selected locus. Depending on the allelic state at the selected locus, this particular segment will belong to one allelic class or the other, right? So think about that population subdivision. So then, say it belongs to A1, you trace it back a single generation. Well, the segment can mutate to the A2 allelic class. Well, that depends just on the mutation rate, right? So it's the probability that mutates times the frequency of the other allelic class. Mutation from that class to this one. It's like this is analogous to the backward migration rates I showed before, right? So the mutation rate here would be the sort of rate at which you go from that one to the other. You just invert that. What's the probability that you came from that, right? Then, of course, you also have recombination with some probability. And otherwise, if none of these happens, it just remains in its class, right? So now we're just following one segment. If there was a recombination event, we have to think about the genotype of the individual in which that recombination event took place. Okay, the lineage is A1, so the individual must either have been an A1A1 individual or an A1A2 individual, and that you can just get from like regular Hardy-Weinberg thing, modulo selection coefficient here, if there's some kind of selection maintaining this polymorphism. Yeah, and basically this is basically, if you assume that the selection is quite weak, it basically is equivalent to the probability that it meets the other allele, right? If selection is relatively weak. Okay. If recombination occurs in homozygote, then both branches after they split this up will remain in the A1 allelic class. If it occurs in a heterozygote, then depending on where that, the bit where the recombination occurs, the bit that carries the selected locus will remain in the same class. The other one jumps into the other allelic class, right? So this is just a bunch of bookkeeping to keep track of what actually happens here. And then coalescence occurs as usual for structural models within each allelic class. Okay, so what do you expect to see? Well, that's intuitively here, right? If mutation rate between these 2 allelic classes is very low, and selection maintains them. So think about that HLA example I was talking about, right? If these alleles are very, very old, then if you happen to sample individuals that belong to the different allelic classes, they can't coalesce until there's a mutation at the particular site, right? And that could be 4 times 10 to the minus, that can be 10 to the minus 9 mutation rate or something like that. That can take a very, very long time, right? So they would just be there. So the coalescence time at the locus itself, so here I basically simulated, okay, so at the locus itself, you expect to have a genealogy. So here I simulated something where 1, 2, and 3 were sampled in one allelic class, 4, 5, and 6 were sampled in the other. And they all coalesce within each allelic class, within each subpopulation, right? But then until they coalesce with each other, it's a huge coalescence time here, right? So remember that the expected coalescence time is like, close to 2, right? In this model. Or, yeah, that's the expected value. But here you have a value of 24, but it's for a very, very small chunk here. So what we're looking at here is chromosomal position. The selected site is here, just at 0.5. Just around that, there's a short segment where the coalescence depth is extremely deep. If you go out then a little bit here, there was a crossover, a recombination event. There were several of them here, right? And you have like a different coalescence time, and then it goes down, and here it gets older again. And as you move away from the selected sites, now you don't necessarily get them all coalescing in the same allelic class anymore. So here, like 2 and 3 coalesces together, but 1, for some reason, recombined over into that class early on, at this position out here. And the coalescence depth is still increased over, but probably not significant actually, over background. And here it's lower, and here you have a different kind of topology, right? So what you'll tend to see is that here the balancing selection will, the closer you get, because recombination gets rarer, the more likely you are to get like a pattern where you divide it into 2 allelic classes and get a deep root splitting these things, right? And this gives a pattern of selection that you can detect. You'd expect there to be like extremely high polymorphism in this region. You expect there to be like clear separation into these 2 allelic classes and stuff like that, right? And so these models were invented to look for this. So do you ever see this? Well, yeah, you do in the MAC region, right? So this is just a so in the MAC, you see nucleotide diversity here. Nucleotide diversity in the human genome typically is on the order of a fraction of a percent, right? It's like 0.1%. In the MSC region, you have like 10% nucleotide diversity. Now, this is cheating slightly because the MHC has multiple sites under selection. I mean, it's a and there's suppressed recombination that causes the whole thing to go up. Other cases where they're probably, in most outcrossing organisms, this kind of region here will be so small that it really will be almost undetectable. There's only a slight blip of increased variability. In selfing organisms where there's a less effective recombination, you can get a bigger effect. But yeah, so anyway, you can see this pattern, and people look for this, right? Of course, the problem is, anyway, we'll get to that in a second. Okay, so yeah, balancing selection. Okay, right, selective sweeps. Everyone knows about selective sweeps. Selective sweeps, here you go. Here's a bunch of haplotypes with different SNPs on them, right, in a population. These are different haplotypes. Here's a SNP shared by these. Here's another SNP shared by these, and so on and so forth. Here's an advantageous variant. It's beneficial. It sweeps through the population. What happens is that unless there's recombination, everyone looks like this afterwards, right? So what does that lead to? And you can simulate this exactly the same way. So here the population will divide into the green class and the not green class. One of these classes will be expanding and the other one will be shrinking, right? So you have changing as a population size. Then you can apply the same recombination machinery, calculate these equations just like you did before. It's slightly more difficult because it's not time homogeneous, right? But it's very easy to simulate. So if you knew the trajectory of the selected sites, then you can just do this backward in time. So what would you expect it to do? Well, so here you kind of get the opposite signal of what I just showed you with balancing selection. Yes, I think I said that right. So, yes, let me see here. Yeah, okay, so maize and teosinte. Okay, so the text here. So, it's a classical example of a selective sweep. The ancestor of maize, teosinte, looks sort of like this. It has a branched growth pattern, right? It has small kernels. It has kernels that tend to fall off, like seeds should do. with domestication, you tend to select the things that keeps the seeds on the plant so that we can actually collect them and eat them later. Large seeds, we want like large, big ones, right? So all the nutrients go into one big fat thing, right? We don't want all these tiny little seeds. We want big, juicy ones, right? So it turns out that a small number of mutations convert. So this is maize. There's a teosinte branched 1 mutation. If you take a maize plant, take the teosinte branched 1 allele, put it in, you get a maize plant that looks like this, right? Okay, yeah, so in this case, what you get, right, around this region, I should have put that slide in, of course, around this teosinte branched 1 locus, you have a depression in variability, and you also have an excess of rare alleles, right? Because what happens is that you wipe out all the variation, and then variability builds up again, and you get lots of rare alleles and so on. So it looks exactly the opposite from the balancing selection one. Okay, so there was one more thing I wanted to say. So we have balancing selection, we have selective directional selection things. The other thing to think about is purifying selection. So purifying selection, here the idea is you have again this pattern, but now there's a bunch of variants here, and some of them are deleterious. Well, what does selection do? Well, over time, these guys get wiped out. Of course, new one comes up again, so it's a slightly misleading thing because mutations already always happen, right? But basically, these guys are, I can't remember who coined the phrase, I mean, they are the living dead, right? You know that these guys are, even though they're around now and they may reproduce, but 100 years from now, they're not going to be there anymore because probabilistically, these guys have no future. The fixation probability for reasonably strong deleterious allele is very, very low. So they may drift around for a bit, but then they're going to be gone, right? That means also that the haplotype on which they're sitting is also dead, unless it manages to break. It's in a bad neighborhood, right? So you can see this as a metaphor, for social situations or whatever. You just can't get out of this slum you're in, right? You're sitting next to all this bad stuff. Unless you recombine out, you're also dead. And what happens is that okay, so this gets wiped out, and this can under circumstances look like a decreased effective population size. And you can kind of think about it this way. Well, so with a continent island model like this, right? If there's a lot of gene flow in this direction, these guys will tend to be replaced over time, right? Yeah? Going backward in time, if you look at the coalescent here, all the lineages you sampled here will tend to go back here. And you can now just replace this by sort of wild type allele here, and here are all the alleles that have deleterious mutations, right? They also have no future, right? So in their entire space will sort of be wiped off over time, right? And this, because selection is relatively quick, you can apply that theory I showed you about sort of fast migration limits. And basically what this ends up looking like is a sort of a source sink environment with quick migration between allelic classes because selection is relatively strong in the coalescence timescale. So that when you crank the math on this, you can sort of, it kind of looks like basically reduced effective population size because over the coalescent timescale, only the mutation-free reproduce. Right? Now, this is of course on a sort of on a very local scale because recombination churns this thing around, and it gets a bit complicated to work out the math in this. But you basically get a model where the extent to which the effective population size is reduced depends on how many deleterious mutations there are per recombination events, as it were. Right? If there's a lot of mutations, little recombination, then you depress the effective population has a lot. If there's a lot of recombination, it doesn't matter. Then it's like you have a lot of social mobility, right? You can move around from everywhere and so on. Yeah? Okay. So to sum all this up, this conditional structured coalescent is very useful for providing intuition about what we might expect to see in the vicinity of a small number of strongly selected sites, right? So people have used this a lot just to see, okay, what would you expect to see? Then these ancestral selection graph models have really not been terribly useful at all, right? Because they're just too hard and it's very difficult to do anything serious about this. there has been some recent work which I've not kept up on. I mean, so something that we really would like to understand better is what happens to the pattern of variation in general if there is a lot of, there's purifying selection everywhere, right? I mean, but these last models of background selection are being used, but typically people just think about it as a decrease in effective population size. So for instance, it is clear if you go across the genome that the level of polymorphism and coalescence times tend to be reduced in genic region compared to outside genic regions. And it sort of fits just like as if the effective population size varied as you go along the genome. Okay, good. I think yes. So are there any questions about this? Because then I'm going to switch to talking a bit about what people do with all this stuff. Right. So again, right? So you were asking Carita before, like, so people mostly simulate neutral coalescence, right? But it's to get like a null model, because the whole point is that almost all the analysis we have assume that almost all the SNPs we're looking at, right, are neutral. So we want to understand how they behave, then under influence of selection and things like this, right? Nobody has really taken seriously thinking about, okay, what should we do if everything is under selection? Because then what do we do, right? Look for a different job, I think. Okay. Then I'm going to switch to, I think I had this in a, yes. Right, okay. So now, what is the coalescent good for? Well, as I've just said, it is extremely good for intuition, right? People generate tons and tons of sequencing data, and if you understand a bit, understanding, on some level, I can't quite tell whether this is trivial or not, right? When I first started doing this kind of stuff 20 years ago, it was clear that most people did not understand it. I think today people, when I talk to my molecular biology colleagues, they still don't understand it, right? But basically, that the pattern you see in a population sample, that there is this underlying graph, that there's like one history that has an enormous amount of randomness in it, is very, very important, right? Then we can quibble about exactly what is the distribution of that object. Now, under neutrality, if you're willing to assume things, you can simulate it in various ways, but you can't get away that it's sort of always there. Like you can go on, clearly people at large still haven't done it. If I go on Twitter, there are all these racists out there who sit around and get Y chromosome haplotype trees and infer, all kinds of things about their glorious ancestry from this, right? And, that understanding that is really sort of a random outcome, that if you actually want to understand something about history, you need to have SNPs genome-wide, so you can actually estimate migration parameters on understanding that this is a much more complicated thing than you think. There are all these companies, some, you can get your genome sequence now, right? And some of these companies are complete charlatans, right, who will tell you all kinds of things that are not true. There's a small number that are quite reputable and will actually give you the story that, nobody ever wants to listen to scientists. We always hedge or think, well, maybe there's, we never give them a clear answer that you are descended from Alexander the Great or something like that, right? Okay, but so that's very important. Now, so other things, so detecting selection, that's still, and we can perhaps have a discussion about why that is the case, but it has been a preoccupation for molecular population genetics for a long time to find genes that are under selection. Somewhat cynically, one might argue that this is why. Well, because at least in the fairly recent, if you had a good selective story, you got a paper in Nature Genetics, right? Because does it actually tell us that much about evolution to find a particular gene that does something? Well, it depends. Maybe it's an interesting gene, right? But as my postdoc advisor, Brian Charles, always used to say, well, what do you need to know? It's DNA, right? It's on some level, it's selection, right? And you find, and I think one can make, and several people have made, a very strong case that the things that you're likely to be able to pick up because they give strong patterns like this are perhaps not typical for how adaptation actually occurs, because most traits are quantitative in nature and changes are not so obvious. But okay, and then the other thing is inferring demography, right? Trying to learn something about the history of a population. And there are parts of this, and I will give at least one example, but there are also some tools that people use almost every day, like that are based on various forms of coalescent theory, like, for instance, tools for phasing diploid sequencing data. And I will perhaps mention that later on. Okay, so intuition, we don't need to say much more. So detecting selection. So, well, okay, so think about it. So what do people do, right? What are the signs of selection in sequence data? If you really want to read any review by like Rasmus Nielsen, if you want a comprehensive thing of this, I'm just going to skim through some of the ideas here. This is not really my field, but basically the signals you can look for are something to do with the allele frequency distribution. And again, the reason I'm putting this up here is that because for all of these, so people look at the allele frequency distribution, they look at polymorphism versus divergence with some kind of other species. They look at Linker's equilibrium of haplotype sharing. And what do they look for in all these cases? They look from deviations from neutrality, and the neutrality you get from the coalescent model, right? That's how it works in all these cases. So for instance, if you look at the allele frequency distribution, under a standard coalescent, and this can be another exercise, but it has to do with, like, if you think about the branch lengths of a coalescence tree, you always have, so if you think about derived mutations, like new mutations, if you polarize them with respect to an outgroup, right? So you have SNPs, you figure out which allele, so you look at human polymorphisms, right? You look at the chimp version and you see which one occurred in human lineage. And most of them are going to be rare, and there is like half as many that are present twice, and then sort of, this is this 1/j distribution that I showed a couple of times, right? So, you know, 1, 1.5, 1/3, 1/4, and so on. And they go like, and they become rarer and rarer if you go out like this. So this is the under neutrality. Okay. And so that's what you expect. And I already told you that with demography, you can change this, right? If there's been an expanding population, you will tend to get more rare alleles. So you skew this entire distribution towards rare ones. If the population is subdivided, you will tend to get more high-frequency alleles because they sort of reach high frequency in different subpopulations, right? Basically, you can get almost anything to happen with population structure, which is a problem because what people also want to do is look for selection, because if you have negative selection on a bunch of SNPs, so you would expect there to be also things pushed towards rarer alleles. Sites, and you can see this in genome data for sure, right? Like if you compare fourfold degenerate sites to non-synonymous sites, the non-synonymous sites will always be skewed towards rarer alleles. This is absolutely true, so the theory works. What usually doesn't work is that it fits a simple coalescent model, but I'll get to that in a second. If you had sites that you believe somehow were under positive selection, you would expect some of them to reach high frequency, right? Because they're positively selected. And if there'd been a selective sweep, like I mentioned, you could get a funny pattern where basically some haplotypes are driven to high frequency and therefore drag along some alleles to high frequency, right? But you also depress variation, and as that builds up again, you get sort of things at rare frequency, okay? Polymorphism and divergence, there are many different versions of this. The most famous one is probably the McDonald-Kreitman test that basically utilizes simple facts from neutral theory that the level of polymorphism should be proportional to the divergence from an outgroup under neutrality, right? This is just a coalescence tree between species and a coalescence tree within species, right? And think about it, there are the branch lengths, and you just throw on the mutations, right? And if you then compare different types of sites here, synonymous and non-synonymous, and if you look at differences between species, compared to the polymorphism, and what you expected to see here, this is an old textbook example, is that in the polymorphism data, overwhelmingly the polymorphisms are synonymous, whereas if you look at species difference, there are many more non-synonymous. And the classical interpretation with this would be like some of these must have been favored by selection, right? Because under neutrality, they should be completely proportional, right? So here, 29% are non-synonymous. Here, it's only 5%, right? And they should be exactly the same. So it could be that, but it could also be that sort of the strength of purifying selection has changed, right? There was bottlenecking during the speciation so that these things manage to reach higher frequency because of that. Okay, and again, and then haplotype sharing. So under neutrality, the frequency of an allele reflects its age, right? There's a classical paper. I mean, you can basically show that the probability that an allele is, because by symmetry in a constant population, the probability that an allele is the ancestral one is proportional to its frequency. That's a symmetry argument that is kind of cute, follows from quadratic theory and stuff like that, right? So in general, an allele, a derived allele that has high frequency will be old, simply because it takes a long time to reach a high frequency. Okay, now, if the extent of haplotype sharing, the extent at which it conserves its original haplotype on which it arose, will also, if it has been in the population a long time, it has a long time to recombine with everything else, right? Whereas a new allele, sits on a unique haplotype. Okay, selection can disrupt this relationship, because if you now take an allele that has been driven to high frequency by selection, say a selective sweep, one way you can detect that is that compared to ones that actually had to drift all the way up to that high frequency, the selected one was basically pulled through the population very quickly. And selection has to act on a log n time scale, drifts act on an n time scale, right? So there just hasn't been time to recombine, so it will drag along a long haplotype, preserving a sort of the one on which that mutation arose. So you can look for that, right? And there's this is G6PD. There's a classical, one of the early papers that started looking at this stuff. Okay, how do we read these plots? They're trying to test for selection graphically here. So G6PD is involved in malaria resistance, right? And so there's a particular allele that leads to G6PD deficiency, but it also gives resistance to, one form of malaria, I can't remember, it's Purifying, I can't remember. Okay, so what are they doing? They take, so here's the site that they think is under selection, and what they're noticing, so you basically, here's the actual site, then you go to the first SNP, and you see how many of these guys that carry this particular allele, that they think is the selected one, are one type versus next. And it turns out that here, there was a different SNP here, but it's very rare, right? So you make a thin line, right? And whenever you run into a SNP, whenever you have a SNP, right, you bifurcate the sample, right? Is that clear to everyone? You start with a haplotype with a particular allele. You go to everyone who has that, there are like 5 individuals that have a particular SNP. You go to the next SNP, right? If that SNP, in that allele class, if that, some of them will have a 0 and some of them have a 1 at the next SNP, that bifurcates that whole sample, right? Okay. Continue. Next one, a next SNP, and the next SNP comes up, right? If there are 2 different alleles here, then you split the branch again, right? And this way, what you get is as you move away, you successfully split it into finer and finer things, right? And typically what happens, if there's a lot of recombination and so on, you get something like this. You start with a particular allele, okay? And the first SNP you run into, right? So basically, most of them ended up having this allele here, right? But very quickly, it's split into, you basically randomized over all the genetic backgrounds that exist at each of these SNPs, right? Okay, but what you're supposed to see here is that in this case, there is an uncommonly long, fat haplotype here, as if, and the invitation would be that this guy has been under selection. Basically, it sort of reached a high frequency very quickly and sort of distorted this relationship. So there are many, many ways of summarizing this. There's an extended haplotype test. There's the IHS. There's like software you can download to do this. People routinely do scans across the genome this way. Okay, but now, if you take the history of this, right? So when much of this theory was developed, the basic algorithm went like this. You sequence your favorite genes. Remember that this was when it was really hard to sequence things, right? Okay, you calculate the test statistic, then you run some coalescent simulations, and or you can compare it to like the, to see whether this is significantly different from neutrality. You can even do this analytically if you're really clever, and if it's significant, yay, you publish, right? Okay, so people did a lot of that. That was like 10 years of papers of this ilk. And a lot of that came from the Drosophila world, where they rarely found anything significant. But certainly when people started looking at plants, and this particular, well, the Arabidopsis work, but also people in maize, when they started doing this on a couple of genes, they're, wait a minute, you know, the Drosophila guys never found anything under selection, right? So they were only the lucky ones that got published papers. But in plants, everyone gets to publish a paper, right? What's going on here? Because basically you ended up rejecting the null hypothesis all the time. So I think we were one of the first who actually did what you obviously had to do. Well, let's test this, right? Not sequence my favorite gene or 2 or 3 genes, like sequence 1,000 genes. And look empirically at what is the distribution of these statistics. And then you see things like this, right? So here's Tajima's D, which should, as if you remember, Tajima's D should have a normal distribution with mean 0, if it's correct, right? If the actual empirical distribution in the genome looks like this, right? So obviously, any test that was based on like putting the 5% tails out here, you know, standard textbooky statistical thing, would be sort of massively anti-conservative, right? In the negative end, to be sure, right? You know, there's like, you know, most of the population is out there, but it's also over-dispersed in the other direction. Here is also, by the way, here's the number of segregating sites per, if you take the average number of segregating sites, run coalescent simulations with that, you would have expected to see a distribution over each locus that look like this. Again, you have something, so this has the same mean as that, right? This doesn't even have the right mean. This is the same, these 2 distributions have the same mean, but the distribution is completely different. So what's going on? Well, obviously, right? It's population structure. The null model, this approach doesn't work, right? Here's another examples that were, I shouldn't pick on these guys too much, but I will anyway. Because there are many papers like this, right? The CCR5-32 AIDS resistance allele was found to be resistant to AIDS, give resistance to AIDS, and it was in vogue for a while to speculate that all these things that were resistant to these things must have been involved in being resistant to something else in the past, and that's why they had such high frequency. So they looked at haplotype sharing, and it seemed like this one had a long haplotype, but you know, it couldn't have been selected just for AIDS, right? Because AIDS was a new disease. So why was it there? Why did it have such a? Why did it look like it under selection? And so they were speculating, and this was a Nature paper that it could have been bubonic plague. This was involved with. Okay, and a lot of us at the time were just facepalm, right? Because you know, not believing it, right? Because you just, again, look, you have no idea whether you're basically comparing it to a theoretical null distribution. You don't know whether this is true genome-wide, right? Because people hadn't yet done what I was just showing you, sequenced across the genome to see what the distribution of the statistic is. And of course, this day, people have done this, and like there was a paper Sabeti in 2005 showed that indeed there was nothing unusual at all. The pattern they saw there was just completely compatible with what you see across the whole. There was no sign of any selection. It was just an allele, probably, that happened to have a high frequency and then happened to give AIDS resistance. There's no story or anything like this, right? So there's a moral to the story, right? You know, so just because things get published in Nature, that does not mean that they're in any way right. And this is one of the rare cases where one of these, otherwise it's a pretty safe way. You can build a career with these kind of things, right? Because nobody can ever prove you wrong in the statements about what happened in history. But in these cases, you actually can, right? Because okay, so yeah, this approach is of course dead. By almost 20 years ago, it became obvious that there was way too much selection this way. And if you think about it, right, in what other field of science would experiments without a control be expected? Because that's really what you're doing here, right? And in the same sense, so now I'm going to recite the philosophically here, kind of what happened if you look at the history of molecular population genetics. There was this Allele era I was referring to back in the 60s and 70s, which had really crude marker data, right? These silly Allele people ran out. You don't know what's going on at the molecular level, but it was cheap. And towards the end of this, people got really good at it, and they had really large sample sizes. And so when they found population structure, looked at this, that, the other, people were trying to infer human ancestry and so on. Then in 1983, Marty Kreitman published the first sequencing paper, right? You know, sequence ADH allele. And at that point, you know, you couldn't get anything published unless you sequenced DNA, right? So then people see, but sequencing was really hard and really expensive. I mean, his first paper had like 11 alleles, right? So then suddenly you got really detailed data on a very, very small region, but the sample sizes were ludicrous, right? So of course, any discussion of population structure like that was, you can't detect population structure with a sample of 4 or 20 individuals, right? You actually need to have a big sample, but nobody could afford to sequence like that, right? You also had no idea what the distribution was across the genome because, PCR wasn't even invented, right? I mean, people used to clone these things and sequence them, and then you had PCR, which is still nonsense, right? But now we have genome-wide stuff, and now it's getting cheap, so, now it's really getting up to like whole genome sequencing and so on, and then things change again. Okay, right, yes. Exactly, and there are papers published on this too, right? I mean, the optimal size for estimating theta is probably 4 individuals, right? But that's a pretty boring question if you think about it, right? That's only if you. So one thing, yeah, it depends on what the question is, right? A problem with sequencing a small number, the neutral coalescent, yes, absolutely, you don't need bigger things. But if you actually want to understand the population structure, which is always there, you need a big sample, you need a geographic sample, so you can actually see the patterns through space and like perhaps get the right model for what's going on. I'm pretty, people did this, right? But it was completely circular. Yes, theory says that in a standard neutral coalescent, we only need to sequence, less than 10 individuals, right? Okay, so we're only going to do this anyway, we can't afford it. And anyway, there's no sign of population structure. But of course there isn't, because you don't have the power to detect it, because you only need 10 things. But theory shows, so like, so you basically, it's complete nonsense, right? And I think this was from Peter Androfato's lab in 2005, showing that, what drove this was what's Drosophila, molecular population genetics. And they insisted that flies had no population structure. But once they got their act together and got bigger samples, it was clear that Drosophila has population structure too, right? Everything has population structure. And you need large samples to understand it. Otherwise, yeah, so basically, this is just showing that the distributions to G - D in largest numbers of loci, I'm being very historical here. You have to, for many of students here, you don't, but if you go back as recently as 2000, it was common that people published papers by sequencing a small number of loci. And of course, in some organisms, people still kind of do this, right? But it's almost unfathomable today, right? Because, yeah, in most organisms now, people do whole genome sequencing, and it's a completely different game. And then you actually need completely different ways of. Yeah. Okay. Right. Okay. So then I'll put that up there for people to think about. This should have been obvious, but wasn't, and there are many reasons why that's probably true. Yeah. So what do you do then, right? Okay. So the problem is, so now if you're going to run this selection scans across the genome, and you know that neutral coalescence will reject things. Basically, you can reject the standard neutral model. There's clearly some kind of complicated demography underlying this, right? So one way to do it, and both have been used in various ways. One is just simply forget about significance, and that's usually the one I advocate. Do not ever trust the p-value. If some editor insists that you should have a p-value for something's under selection, just tell them to, no, it's impossible, right? What you can get is a list of candidates, a prioritization, right? And then you have to come up with some independent way of verifying this. And things one can obviously do, and you can clearly see that there is reality in this. So things that show strong selection signals tend to be things where you have independent evidence that something is going on. Like, for instance, things that are, you can compare genes and non-genes, right? Okay? If you don't see a difference there, you clearly have a problem. Now, in almost all organisms, you do. In human genetics, things that have been implicated in GWAS hits tend to show stronger signs of selection. Things that are listed in the online Mendelian inheritance in man as being involved in diseases and so on. You can use gene ontologies, as you were showing yesterday, and you can see that certain classes of genes behave differently and so on, demonstrating that there's some kind of real signal in this, right? And then what you do further on, that depends on what the question is, right? A problem with this approach is that it does not lend itself to identifying the gene under selection, right? It can only statistically say, well, this group of genes are overrepresented, so something is going on here. but from an evolutionary point of view, maybe that's actually what we really want to do, right? Because we're actually interested in quantitative understanding of adaptation, not just finding a gene that does something cool, right? It is, however, a less successful way for getting high-profile papers, but that's, now I'm being cynical again. You can also try to identify statistical outliers by calibrating the null model, and this is something else people do a lot of. Because remember, the neutral coalescent is very fast to simulating things, right? So if you are willing, if you have some idea of what kind of population structure would have given rise to the deviations you see, you just simulate the heck out of it. I think, did I put, yes, okay. Okay. You just simulate a lot, and you just basically pick values that tend to fit the data, right? And I think I have a slide about that later on. Right, so yes, I already said this. Enrichment tests. Yes. Yeah, right. The problem here is, of course, can you actually get the model right? I mean, so what people actually do, right? You have a large data set, and you can do, you remember those figures I showed you with like Tajima's D in Windows, and then you have the number of segregating sites in Windows, and then you can come, then you can just simulate various models of population growth and subdivision and see if you can get something that fits that model, right? It's an incredibly powerful simulation tool. You can use this, if you want to be formal of it, you can refer, this is known as approximate Bayesian computation, right? Where you actually estimate parameters by looking at proximity to the data. As I've been trying to tell you, if you actually have some idea of what the demography is, it's very easy to model it. The problem is that reality is very complex, and coming up with a parameterization of this that actually works is very hard. Yes? Mm-hmm. Yeah. Hmm. Mm-hmm. So, okay, so... Yeah. So I think starting point is, well, I can tell you, so my version of this would be, okay, I will tell you what I think, but you should probably meet someone more optimistic and have a drink. And you have a drink, then meet someone more optimistic. And I think it's almost impossible, to be honest, unless you have some kind of framework, like, some very clear idea where to start, right? There are just too many things to choose from. I usually don't do it. I tend to ask different kinds of questions. Is that my research has been shaped with this? I grew up doing this stuff, right? But I will show you in a second, like for at least the Arabidopsis data, I have actually given up trying to fit it to models very well, because if you just sit back and think about what must have happened where these guys are, right? Well, I'll show some examples later from some of the work we've done. Where we generate huge amounts of sequence data from a lot of different species, and people want us to estimate all these things using all these models. And I'm like, no. I mean, I don't want to spend my life on this because I don't think it's going to lead anywhere, because effectively, you would have to model something where there have been multiple, there's been divergence, speciation, there has been, several glaciations, which we only partly understood, refugia happening, you need to have accurate climate data. I don't think we're anywhere near understanding this, right? I mean, these historical models, I mean, if you have a very clear hypothesis you want to test, then I think it makes sense, and I will give you some examples of this, right? I mean, for some of the human data, there's a good archaeological or historical record you can use to sort of at least formulate, well, this is the specific question you want to test, right? Sometimes it's the case for some of the stuff we're doing, well, we know that there must have been a migration from here to here, there was a geographic barrier here, so we predict that this should happen. Does it fit that idea, right? But actually trying to, I mean, I don't even know how to, well, it's like I was saying something like that before when talking about migration. I mean, I've been asked by reviewers sometimes to, you should estimate the migration rates, but there was somebody wrote a review read, and I can't remember, it was like, maybe I saw that on Twitter, or someone pointing out like, I mean, if you think about this as an engineer or something, you estimate migration rates, right? What even is the migration? What units do we measure it in? Right? I mean, it's sort of a like, it's like a modeling contrast. I mean, what's the point of trying to quantify something? It's more like, a thought experiment. I mean, you can get a rough idea what could have happened, right? Does it look like population growth? I mean, I'll show you some of this Lee and Durbin stuff in a second, which hopefully, yeah. So anyway, and this clearly is a beer level discussion. So well, let's continue later. Yeah. Don't have a good answer. I don't know. It varies. I've seen all kinds of versions of this. Right? I mean, something people always fit some kind of population growth, kind of, for instance, right? People often fit background selection thing across the genome and stuff like that too. But yeah, people often try to group things into different subpopulations, which is very dangerous because whether that's a good model or not, because in reality, what you probably had is some kind of isolation by distance, right? Because that's usually what you have in nature, right? And I don't even sure how you would really parameterize this. So yeah, I don't have terribly, it's like, yeah, I think the first thing is what's the question actually, and what are we actually trying to answer? Okay. Yes. Yeah. Oh, yeah. I was going to say so. Arabidopsis. Yeah. So, I just wanted to show you this thing. All right. So, yeah. So we played around with this. I had some really smart people in my lab who were trying to do this. We never found anything that fit. So here's one thing that I still don't really know what the answer to is. I've been meaning to write a paper at this for a long time now. If you look at the level of polymorphism across the genome, you can see here that it goes up tremendously around all the centromeres. Okay. And we still don't know what's causing that. Right? I mean, one logic that is in this model fitting stuff is that somehow the genome all follows the same distribution except where there's selection, right? Okay, so these isolated selection things, but this is clearly showing you that there's something very systematically different going on along the whole genome. I think the genome is actually organized in ways so that genes that are under different types of selection are sitting in different types of the genome. And we need to model that as well, right? But even taking that aside, if you just the variability here, we were really never able to find any kind of model that even, so you were asking about parameters. So we look at, you look at 3 parameters, you fit it, right? And then, but what if you had a 4th one? It doesn't fit at all. You fit that one, right? And you add a 5th one, that doesn't fit at all, right? And then you iterate until you find something better to do with your life. Sorry, I'm being negative here. Okay. Yeah, okay, so what else do people do? There's a lot of testing for local adaptation, right? Classical papers, Lewontin and Krakauer, who was sort of, this was revived by Josh Ake, quite a long time ago. But there's a lot of stuff going on about looking at correlation to the SNPs and environmental variables. So here's something, one paper that I happen to be involved with. Basically, this was, so, you can do this fairly rigorously if you have field experiments. So this was Annie Schmitt who took accessions, sampled from all over the place here, and grew them in 4 field sites across Europe. And then looking at which alleles sort of were correlated with high fitness in each of these things. And what you saw is that the alleles that were correlated with high fitness tended to be alleles that had a high had a centroid of distribution close to where they came from, right? Does that make any sense? So you basically, you grow things on field sites here, right? Then this is in Norwich, England. And then you look at the alleles that are associated with higher fitness there by some arbitrary fitness criterion. It doesn't matter, right? Again, I don't believe in the p-value, it's just some random cutoff, right? These look like they're correlated with higher fitness. Then you ask, okay, where are they? These were measured by seed set, right? So these were actually control experiments or common garden experiments. So this you can totally do, right? So this is, this is not just looking at sequence data, it's actually doing an experiment, right? And then you measure fitness, right? So this is similar to what Sandy, this is real fitness measurements. There's still a proxy for fitness, but it's something. And then you just look for each of these SNPs, right? Where is the center of its distribution, compared to the whole sampling area? And what you saw then, right, is that basically the ones that are, it's a pity Ophelia isn't here because she would love this, because basically, yeah, so here, you see that basically the alleles that did better here tended to be localish alleles, right? Same thing for here, but this is why it's a pity she's not here, because basically this might be some of that lag from the, you see the same thing here, right? But basically they're much more south than they could have been. And this could be like that they're not really caught up with the ice retreating, right? Anyway, right, okay, so, but I just wanted to, so there's a lot of things out there where people correlate things to geography, come up with p-values and declare these as significantly associated with this, either by looking at their FST outliers, right? That's one thing people do a lot. They look at FST value, which ones are the outliers? Or these are significantly correlated with environmental variables. And, you know, no, right? Never believe this. Always, you know, I mean, it's like, yeah, sure. I mean, it depends. It's between you and your God, I guess, right? You can get things published this way, right? But, you know, no, it's not calibrated. Typically what it means is that the author tested some trivial scenarios and, you know, yeah, whatever, your statistics seems to be robust to that, but is it actually robust to reality, right? If someone held a gun to your head and said, right, okay, we're actually going to be able to find out whether you're right or wrong, do you still want to publish that paper? Nah, probably not, right? Most people wouldn't do it because you actually don't really believe that this is true. You know, the only way to demonstrate it is having some kind of internal control. So like, you know, so simple things you can do. So this was another paper. This was from Angela Hancock, where we're looking at associations. She found things that were correlated. And what you can see here, all I really want to see, there's an enrichment for various categories. But the enrichment is really only seen for the non-synonymous versus the synonymous sides, right? So here, like, something you can do is, like, you can demonstrate that there is a signal, something is correlated, and it only falls on a certain type of genes, right? Then at least you know that there is signal, right? That doesn't tell you that this particular one is real, but it's telling you in aggregate what you're doing is on some level right? I mean, they can't just be nonsense. Okay, yeah, so this is what I'm going to show you here. Yeah, so right, and here's another statement. You read, no evidence of population structure was found. Don't believe that either. Something people I see a lot is that authors try to infer populations using some statistic, which has little power. They find nothing and then proceeds to use some other statistic without demonstrating that absence of structure with respect to this implies absence of structure with respect to this, right? I mean, you know, something can be in Hardy-Weinberg, you know, Hardy-Weinberg, for instance, is incredibly robust. Almost everything is Hardy-Weinberg. That doesn't mean that the coalescent is going to behave neutrally and so on and so forth. Okay, again, that's what you have to do. Right, so this is my moral of the story. In defense of all my molecular biology colleagues, they would never dream of publishing a paper without having a control, and that's kind of what molecular population analysis have done for a long time, right? Right? Typically, when people, okay, so here's just a random experiment I picked out of one of my colleagues' paper here, this is flowering time in Arabidopsis, you see that there's a different expression of this gene depending on the treatment, right? And to demonstrate that that's true is that then they take some other gene and show that that one doesn't do this, right? This is known as a control. In too many papers in population genetics, molecular evolution basically said, well, theory says that this gene should be behaving like this without actually confirming that it actually do that, right? So please don't do this anymore. Most people don't do that anymore, but you still see it from time to time. Right, okay. Yes, okay, so since I'm being too negative here, so, you know, it is possible now to actually get phenotypes, to actually not just look at sequence data. You can do field experiments to demonstrate local adaptation. And I also think it's very important to say this, right? I mean, all this chasing for a gene that does something. I mean, there's been several good reviews written about this fact that, you know, almost everything we know about adaptation suggests that it's, you know, it's quantitative genetics, right? So if you actually want to understand, you know, you need to understand it on a quantitative level. You know, so perhaps it isn't so important to find the gene that does something. It's to actually understand how selection shapes variation in the genome as a whole. Let me see, how am I doing in time here? Oh God. Oh, I have a few more minutes. Good. Okay, so. Yes, I wanted to say something about gene trees and species trees. So here's something else where coalescence stuff is going on. So I'm selfishly picking one of my, there are many, many things like this. I'm just picking one of ours because I'm lazy and, you know, didn't want to. This is one of my former students. We sequenced the entire genus Arabidopsis So Arabidopsis thaliana everyone knows about, but there's also a whole bunch of other species, and it varies a lot. So Arabidopsis has 5 chromosomes. It's selfing. Most of the other ones have 8 chromosomes, and they're outcrossing. They're also polyploid species. There's a lot of variability to, some are specialists, and they're very drought tolerant. Some of them are specialists for heavy metal soils and things like this. Okay, and yes, I wanted to show you. So remember, I was drawing something with gene trees and species here. So here's a cloud of trees, right? So I can't remember how many genes she filtered very heavily to get genes that you can align across all these things. And what you clearly can't see here, but the idea is, if you take so here are all the taxa that we sequenced, right? And we had several individuals of each, and so on. And of course, we have thousands of Arabidopsis, but we just took a subset here. And by and large, they sort of cluster together into recognizable things here. And you can, but if you take individual genes that we can align and build trees for these, you basically get this forest here. And you can see, for instance, okay, and what these things, that basically, and each of these trees give different histories, right? Not completely all of them. So for instance, here is 100%. It means that basically 100% of these, I think there might be 900 different trees here or something like that, superimposed on each other. Every single one of them showed Arabidopsis thaliana as an outgroup to all the other ones, as is being shown here, right? Okay? And you can kind of, if you squint your eyes, you can see that all these trees have branches that do that here. Whereas in here, it's a complete blur, right? So statistically, you see that these species are well put together. Here are 2 things from southern France that also seem to have quite a lot of support, right? The majority of trees show these guys clustering together. But for the other ones, it's a bit of a mess, right? So this is something, and with these kind of things now, you can, if you want, simulate and try to get some idea of what kind of geographic scenario could have given rise to this. It's complicated though here because we don't have any clear assumption of what could have happened. We tested some particular hypothesis, but okay. So this is something you can do. Yes, and right. Oh, I wanted to show you. Oh, I have to tell you also, so something else that people do a lot is, you guys know what these Abba tests are, the D tests? Okay. Right, so here's a cute thing that can be done here. So so if you look at that tree, there were several very surprising things that came out of this data. That this would be a mess, that would look something like that wasn't terribly surprising. But something that was interesting was, so let's forget about these. Let's think about these 3 big clumps here, which are all outcrossing species, right? Basically, so these 3 kind of, do something like this, right? And they're supposedly separated by maybe 3 million years or something like this, right? And then you have thaliana, which sort of comes in here, and depending on what mutation rates people use, people have said that it's 6 to 12 million years. So this, okay, so this is Arabidopsis arenosa, Arabidopsis halleri, and Arabidopsis lyrata, right? And here is thaliana, right? Okay. Now, so there were a couple of things that were weird. So a very surprising thing that we find an enormous amount of allele sharing between all of these, suggesting perhaps at first that there could be balancing selection, maintaining things, that there is an overrepresentation for various resistance genes, including, for instance, virus resistance genes, that seem to have been segregating across all these species. So that could actually be sort of balancing selection. And that's what we first thought we were going to write the paper about. But then we realized there were way too many, and there was something else that's weird. So there is something, there's something known as the Abba test. So, which was invented in the context of humans to show that we're admixed with Neanderthals. So, okay, let's take the Neanderthals and the humans, right? So basically, so the idea was like, okay, Neanderthals have been separated from modern humans, or have they maybe admixed? We talked about this before. So the way they tested that is that they basically said, okay, if Neanderthals haven't admixed, so here's Neanderthals, whoops, Neanderthals are an outgroup. Here are anatomically modern humans, these guys are Africans, and these guys are Europeans, or Chinese, or Indians. I mean, I think they didn't use Indian data, but whatever. Okay, if Neanderthals are an outgroup, then, then Neanderthals should be equally unrelated to both of these groups, right? Yep. Okay, one way you can test that is by having a further outgroup. So now we bring in the chimps here. And you look at any polymorphism, and there are some of these because these are closely related, where there is an allele. So either this or this shares an allele, or this and this shares an allele, but this one not, right? So you look for patterns where you would have, so it has to be a derived allele. So if this is 0, right? This is 1, so a mutation happened here. Then this has 1 and this has 0, or the pattern where this one has 1, this one has 0, this one has 1, this one has 0, right? Okay, and if there was a clean species split here, then these should be equally common. So, so this is basically the A, B, B, A, or B, A, B, A pattern, right? The Ava pattern for here. And basically, so you just come up with the statistics that just separate these things and normalize by the number of things you see. And what they saw then is that, oops, there's actually much more shared of these derived polymorphic alleles between Europeans and Neanderthals than there are with Africans. So Neanderthals must have admixed with Europeans, which makes sense since they're in the same place, right? Okay, so that's a demonstration that there is no species tree like this. In fact, this was a gradual thing, and these guys were actually admixing here, right? Okay, so we did the same thing here, and what we found was that these guys actually are closer to these guys. All right, that doesn't make any sense, right? Again, these are supposed to be quite separated, and you saw those species trees up there. This thing really, and this, this is a self, this is 5 chromosomes, these 8 chromosomes. They are clearly not exchanging any genes today. You cannot cross these. These guys you can easily cross. There's no wonder that they all blah blah blah, right? But here, so that tells us immediately that something. There has been a lot of admixture here, and probably, and so the weird thing is that given that these only split from each other maybe 3 million years ago, that how can it be more close to one of them and not to the other, right? Somehow, this admixture must have happened here. So this thing must have been, all these genomic changes and so on must have happened much more recently, and there was something going on back here, right? So again, for all this stuff, you can build gene trees. It's very easy to simulate coalescent trees and test various hypotheses. So here's an example where detailed demography perhaps doesn't matter that much. You have a pretty clear idea. You want to test specific things about gene flow between specific groups and so on. And that you can very easily do with coalescent models. Okay, I have a little bit more time, great. So, and then another thing that I think we need to talk about. Right, so here's something people use a lot to infer demography. This is the pairwise sequentially Markovian coalescent model. So this is from Richard Durbin and Heng Li's work. It's a very cool paper in 2011. A lot of people have thought of this before, but they sort of made it real. And there is now, it's been elaborated. So actually don't use this method. There is basically better models being done, but I'm just explaining the original one. So the idea here is that, okay, if you even just sequence a single genome, you can look at the pattern of a single genome, right? So now we're talking about diploid individuals, and you can just look at the heterozygosity as you go along. Right? You sequence me for any site in the genome, right? I'm either homozygous or heterozygous. And that reflects basically the coalescence time in that bit of the genome, right? So if there is a, so now we're thinking about the coalescence tree for a sample of size 2, right? There's only one topography, right? Because, topology. The only thing that differs is how far back, what's the coalescence time? And that should be exponentially distributed, remember? So the process that describing this, we know what underlies this, is a process like this, right? The time to the most recent common ancestor would go like this, and then here, there was a recombination point, and suddenly you switch to a deeper ancestry. And when there's a deeper ancestry, you should have more heterozygous sites. And then there's another crossover point, and you go forward again. Now, importantly, if you remember I talked with this ancestral recombination graph, there's, of course, lots and lots of recombination events here, too, that end up not changing the coalescence time depth, right? Because things just coalesce back together again and don't matter. And I'll get to that in a second. Okay, so thinking about this is like really cool. What if we could, what determines this, right? You can basically estimate the time to the most recent common ancestor. And you can look at the length of these fragments, right? And you can basically see how the coalescence rate has changed through times by looking at sort of long, recent long fragments where there is not much going on and so on, right? You can basically look at the whole distribution of our coalescences, because if you go across the genome, there would be hundreds of thousands, perhaps even millions of these, right? And you can basically estimate how this has changed over time. Okay, what do you do to do this? Well, this, if you think about this here, this is kind of like a hidden marker model, right? You basically have some kind of hidden states here that emits heterozygotes or homozygotes, and you can calculate what that rate would be conditional on this thing, right? So if you can just describe this process, you can code an HMM that runs along the genome and does this and basically outputs all these parameters. Only problem is that the ancestral recombination life, as I described it. So what you're doing then is you're walking through tree space. Remember, I drew this complicated graph and then said marginally, there's a tree at every point. Those trees change because of recombination and coalescence. They will be correlated as you go along. It's like, wow, cool. You can instead of doing this coalescent graph thing, you can just have a Markov chain that runs along the genome, except, unfortunately, it is not Markovian because, the ancestry here depends on trees a couple of steps back, right? The Markovian process is that the next step only depends on where you are now, right? But a couple of people, and I put those papers, so sort of, if you want to read more theory stuff, came up with an approximation and showed that it was actually pretty good. You can almost ignore that thing. So you can actually do this kind of thing. Instead, you have a process where you basically figure out where was there, so at some rate, as you go on the genome, recombination happens. When they happen, you basically sort of erase the branch above that, and then sort of attach it according to the length, the total tree length and stuff like that, attach it somewhere else, and you get a new tree, right? And then you get a much simpler process where you actually, you get a Markovian process that goes along the genome, and it turns out that that's a pretty decent approximation for the standard coalescent. Having done that, then you can estimate how effective population size has changed over time. You can look at when back in time was there a lot of coalescence, because now you have lots and lots of these chunks, right? And you can see how old are they, what's the distribution of that, and you can infer, remember that analogy of that box of beetles, how that has changed over time. And this is from this original paper. If you do that on human stuff, you see something that kind of makes sense, that right now, the effective population size is extremely large, coalescent rates is very low. Well, there's a lot of us, right? And we spread all over the world. But if you only go back here to within the last 100,000 years, there was a big dip in population size. And if you look at the colors here, so these are Yorubans, these are Europeans, these are Koreans, and these are Chinese. And you can see that the dip was much less pronounced for the Africans than for the out of Africans. So this is, this is that out of Africa demographic event, right? Because, a massive transition, massive migration from Africa that did a lot of things, then population goes up again and changes, and what happens back here, nobody knows, because, once you go back in time, everything has coalesced, there's no information anymore. And Okay, and you can also, another thing you can do, and this is kind of something that is actually very informative, but you have to have, okay, you can do things like that. You can, so this is just looking at heterozygosity within individuals, right? But if you actually have haplotypes from different populations, you can compare, okay, what's the coalescent rate between different groups? How has that changed over time, right? And, then you see here that there's a bigger split between Yoruba and Africa, and so on and so forth. Okay, now, so there's been a lot of, these guys did it exactly right in the paper. You should read the supplement. They're very clear of the fact that they're not actually estimating population sizes here, right? You're estimating the coalescent rate, right? As we have said, for instance, when things go up or down here, that doesn't mean that the numbers of individuals go up and down. It could mean that there was more subdivision in the population for various reasons, right? For example. And so, but, we've used it, I think it's used very routinely, and it's a very cool thing to do, because it gives you some idea of how things coalesced, right? And then it's up to you to sit down and think, right, well, what could that have been? Why is that? Why, there seems to have been a lower rate of coalescence about 150,000 years ago. Well, what do I know about the climate pattern? It's like any other kind of modeling, right? You can summarize the data in a cool way, and you get information. If you do something sensible with that, it's sort of up to you. Okay. Right. Okay. And I think that was all I was gonna say. And actually, in this context, that simplified. Another tool that many of you will run into, if you haven't, is phasing of data. You're all familiar with this problem? Like when you sequence a diploid individual, right? So here's a SNP. It's heterozygous here, so sequence me. Here's a SNP. I'm heterozygous. There's a 0 and a 1 allele, and there's a 0 and a 1 allele. But you have no idea of whether the 1 and the 0 are sitting, the 1 and the 1 are sitting together on the same chromosome, or the 0 and the 0 are sitting together. But you can kind of see that you can use a population genetics model to try to infer that, right? Because if you have a large sample, you will see the homozygous individuals in some other lines and so on, right? And you can basically use that information plus the coalescent prior to try to reconstruct the haplotypes based on diploid data. And they're extremely powerful algorithms that are being used every day by lots of people that do this. And they all use a coalescent model. Again, not the full coalescent model with the recombination because it's too complicated, but another is approximate simplified things, right? So this, this is being used. It works, right? This is something you can test, right? Because you can actually then use pedigrees or whatever, right? And you can actually see, wow, we actually got it right. And as rare haplotypes you never get right because you haven't seen them before, but for inferring, so this is a very practical use of coalescent models that is being used all the time. Okay, now I'll be quiet. So. And I'm even on time. How about that? Any questions? Yes. Yeah. So this is just looking at the rate at which coalescences occur. So which you infer from basically, you have a distribution of coalescent events, right? Time to most recent common ancestor, right? So if you estimate, as you go along the genome, sorry, I didn't make it speak again. So as you go, what this algorithm does is it goes along the genome and it estimates how the time to most recent common ancestor changes along. So if we sequence you just start from one end on each chromosome, right? You just go along and you estimate, okay, there was a common ancestor, say 400 years ago. That's very rare, but that's going to happen. That's going to be a long chunk of things, right? Then typically you get like, in recent time, right, you get long chunks, right? Because there hasn't been so much time for recombination. There are going to be some bits that go back very far, most coalescence time in humans is on the order of hundreds of thousands of years, right? So you get, chunks like this. And this is 200 kb. The human genome is, what, 3 gigabases, right? So you have a lot of these data. So you get an intensity of, you get a distribution of when things coalesced. And that gives you basically the intensity of how much coalescence was there at any point of time. And the rate of coalescence is depending on this effective population size, right? So, if you basically invert that rate, I mean, I would plot the coalescence rate directly, but people prefer inverting that thing and thinking about it as the effective population size instead. Nope, it does not. It means, so, you're not here for the lecture on Tuesday, I think, but it means it's related to how many people there are, it's related to how subdivided they were, it's related to age structure, it's related to variance in reproductive success. It's a compound variable that bakes into all these demographic things. It basically governs the rate of drift. And we call this effective population size. It is surely related to demographic numbers, but not in any linear, obvious fashion that I would believe in, right? This is based on human genome data, yes. This is the HapMap data, human HapMap data. So, yeah, so when this is, sorry, yes, this is human genome data. They had 96 individuals for the first HapMap, right? And they had, so this is one Yoruban individual. Yorubas, a tribe from Nigeria. This is one European, second European. Europeans actually were from Utah, but whatever, they were white. And then a Korean and a Chinese person, right? And you can do that. This is years into the past. So this is 10,000 years, 100,000 years, a million years. It's the same scale. Here's the axis, right? This has to do with rescaling it. To get this axis right, that's inferred from mutation rates. You have to make assumptions to that. This is just looking at going backward in time, and what you're seeing here is how the effective population size changed. And what you get from this is that there was, relatively low rates of coalescence right now, and that fits because we are an enormous population, right? This is more recent. This is now, and this is way back in the past. And since everything. Mm-hmm. Exactly, and that's expected, and that's actually that doesn't mean that the model is right. That means that coalescent theory is right, because basically, if you go back this far, we all have a shared history, right? If you go back 100,000 years, almost all the genetic data is in Africa, and most of the polymorphism that is back there is shared between. So it doesn't. Whether these actually curves are correctly estimated or not, I have no idea. But they should be agreeing with each other. Because it's basically we're estimating the same thing, whether we're estimating it the right or wrong. Where you see, but the interesting thing is where they diverge, and they diverge in sensible places. About 100,000 years ago, there is a divergence between Africans and out of Africans, right? This was in well, almost every paper that does genome sequencing uses this algorithm these days. It's got to been, this is Nature 2011. But, we have used it. We used it in a recent paper on vervet monkeys. But almost any paper these days that sequence entire polymorphism data sets from some natural population will use this thing, right? So it's because it's a very cool tool, right? I mean, and if you, again, it's a good way to summarize the data, and if applied with taste and a little bit of understanding what actually goes into it, then you can actually learn a lot, right? And you can also make some silly statements if you don't really understand what goes into it, right?